I'm happy to share that our next honorable speaker, Professor Kunal Roy, has joined us. And the topic of his talk is in silico toxicology using QRASAR and ARKA approaches. But before we begin, it's time for a grand welcome for him. So once again, I would like to invite the co-coordinator of this FDP program, one of our finest researcher and associate professor at Dr. Sintu Kumar Samanta. Sintu sir, please give a warm welcome and a brief wonderful introduction of Professor Kunal Roy.
Okay, thank you, Sarfraz. It's my great pleasure and honor to introduce our distinguished speaker, Dr. Kunal Roy, professor at the Theoretics and Cheminformatics Laboratory and former head of the Department of Pharmaceutical Technology, Jadavpur University, Kolkata. Dr. Roy is a highly respected name in the field of medicinal and pharmaceutical chemistry, especially known for his pioneering work in quantitative structure, activity relationship, QSR, and molecular modeling. He has an outstanding academic background, being a gold medalist in both B.Pharm and M.Pharm from Jadavpur University, and later completing his PhD from the same institution. Over the year, Professor Roy has earned numerous prestigious fellowship and awards, including the Marie Curie International Incoming Fellowship at the University of Manchester, the Commonwealth Academic Staff Fellowship, and the AICT Career Award and the DST Fast Track Young Scientist Award. His research contribution have also been recognized internationally with several most cited paper awards from Elsevier journals such as Bioorganic and Medicinal Chemistry and Bioorganic and Medicinal Chemistry Letters. He also served as the co-editor-in-chief of Molecular Diversity Springer and is a member of editorial advisory board of reputed journals including the European Journal of Medicinal Chemistry, Journal of Molecular Graphics and Modeling, Chemical Biology and Drug Designing, and Expert Opinion on Drug Discovery. Dr. Roy's research interests cover a wide range of topics from studying the QSR of pharmacologically active ligand and antioxidant to modeling physiochemical properties, agrochemicals, and environmental pollutants. His work has a remarkable impact on drug discovery and computational chemistry worldwide. We are truly privileged to have such a renowned scientist and academician with us. On behalf of everyone here, I extend a warm welcome to you, sir. We eagerly look forward to hearing your valuable insight and learning from your past experience. Over to you, Dr. Roy.
Thank you for a nice introduction. Am I audible?
Yes, you are.
Yes, sir. Yes, sir.
Okay. And I think you can view my slides. I have already shared.
Yes, sir. Yes, sir. Yes, sir. Yes.
The topic is, as you already know, that is quantitative read across structure activity relationship and ORCO approaches with reference to in silico toxicity prediction model development. Now, to start with, we are surrounded by a large number of chemicals. We are exposed to chemicals from our everyday uses of different kind of compounds, starting from the household uses of, say, biocides, pesticides, cosmetics, personal care products, pharmaceuticals, and other industrial products. The effect of hazardous chemicals and pollutants on the ecosystem is a matter of great concern because although we have a large number of chemicals in the environment to which we are exposed, relatively few of these chemicals have been subjected to adequate assessment for their hazardous environmental properties. Now, with the ever-increasing production of new chemicals and as the cost of experimental testing is rising, and also we have to consider the requirements to optimize resources for risk assessment of existing chemicals in use, the regulatory agencies from all over the world, for example, the US EPA, OECD, ECA, European Chemicals Agency, etc., recommending alternative methods, including in silico approaches for ecotoxicological risk prediction. So as we have to give more emphasis on alternative methods to in vivo toxicity testing, as we have to reduce the animal experimentation. Actually, the animal experimentation is banned for some special kind of compounds. For example, cosmetics. For testing cosmetics ingredients, we cannot do animal testing. We have to rely on either in vitro or computational approaches. Under the REACH regulation, registration, evaluation, and authorization of chemicals, which is effective in European Union, more efforts are being given to include methods such as in vitro techniques and computational methods, including QSAR or quantitative structure activity relationship models. So these kind of alternative approaches are termed as new approach methodology. These are time and cost effective. This avoid animal experimentation. They support 3R principles: reduction, replacement, and reduction of animal experimentation. They can be applied to virtual compounds, and they are also supported by various regulatory organizations. Now, QSAR is an acronym of Quantitative Structure Activity Relationship, which deals with development of predictive models that correlate biological activity, which may be therapeutic or toxic, of chemicals, which include drug, toxicant, and environmental pollutants, with chemical structure information, which is mainly represented in the form of descriptors. So descriptors are nothing but numerical depiction of chemical structure information. And this kind of relationship is developed by the application of statistical tools. So the basic notion of any QSAR is that biological activity is a function of chemical structure and or property. Or in other words, we can say that biological activity is the function of descriptors. Now, if we want to see some simple examples from drugs, we can give examples of some phenothiazine drugs like chlorpromazine, promethazine, and ethopropazine. Now, chlorpromazine is an antipsychotic drug, has a side chain like this. Now, in case of promethazine, there is some chain in the side chain. As you can see, there is a branching. And in case of ethopropazine, it's an anti-Parkinsonian drug. There is a diethyl substituent. So small changes in the substituent may change the therapeutic category. Again, tolbutamide and chlorpromide, they are structurally similar. They are sulfonylurea derivative. Now, tolbutamide is hypoglycemic, whereas it is short acting, and chlorpromide is longer acting. This occurs due to presence of the chloro substituent in chlorpromide. We can give other examples like isoproterenol and epinephrine. Isoproterenol is hypotensive, whereas epinephrine is hypertensive. And this occurs due to changes in the substituent on the nitrogen. In isoproterenol, it is isopropyl substituent instead of methyl as in epinephrine. So as you can see, with small changes in substituent, the type and magnitude of the biological activity may change. Not only changes in the substituent pattern, but also their orientation in space is also important. So stereochemistry is important. For example, diethylstilbestrol in trans form is more estrogenic. Similarly, propranolol is effective in the laboratory form. So here only the hydroxy group orientation space is changing in propranolol structure, and that's why one form is active and another form is inactive. Similar case occurs in case of epinephrine, where minus laboratory form is more active. So stereochemistry is also important. So with changes in the substituent pattern, with changes in the structural features, with changes in stereochemistry, biological activity of a series of compound changes. And these changes occur in a very systematic way. And we can identify the pattern, and we can encode that pattern in the mathematical format. We can develop some models. The models can quantitate. And for developing such models, we should have a set of compounds with known the biological activity or some property values, and based on that we can develop a model, and once the model is developed and properly validated, we can apply this model on new set of data for prediction parts. That means for compounds for which do not have experimental activity values, we can fill the data gaps by using model derived predictions. And for data gap filling, we have different approaches like expert-based system or QSAR, data cross approach, which is basically a similarity-based and expert rule-based approach for structural alerts. So today we'll now we should information. Chemical structure information can be encoded in different way. They can be encoded in the form of molecular graph. They can be encoded in the form of molecular descriptors, which are numerical quantities to describe molecular structure information, or we can encode them in the form of fingerprints, which are mostly presented in binary form. We can also textual representation of chemical structure in the form of smiles or image. Now, depending on the chemical structure representation, we can get molecular descriptors in different level, and molecular descriptors of different level have information of different dimensions. Now, from molecular formula and one-dimensional structure, that is the textual representation of chemical structures, we can get constitutional descriptors. From two-dimensional representation of molecular structure, we can get topological descriptors, structural, functional group counts, etc. From three-dimensional representation of molecular structure, we can get geometrical, electronic, and spatial descriptors. From surface structure representation, we can get molecular shape analysis descriptors. And sometimes the ligand-protein interaction can also be used for computation of descriptors, but mostly for QSAR we use only ligand information, not the information from the target. So QSAR is essentially a ligand-based approach. So starting from appropriate molecular structure representation, we can get some numbers which can encode molecular structure information. So we can give some simple examples of molecular structure information, like say partition coefficient or log P value, molecular weight, molecular refractivity, pKa value, that is the acidic dissociation constant, and some simple structural information like presence of aromaticity or number of double bonds, number of carbonyl information, but quantum chemical descriptors. So we can calculate a large number of descriptors. So these descriptors are usually presented in the form of columns, and the compounds or data points are presented in the form of rows. So we'll have several rows. Each row corresponds to a particular compound, and we have several columns. Each column corresponds to a particular descriptors. So we will have a large table, but we have to reduce the dimension of this table because there will be some redundant information. Some of the columns may be intercorrelated, or there will be some of the columns which may have no variance in their values. So you have to omit such columns. And also you have to consider that all columns or descriptors are not important for the endpoint or biological activity that you are going to model. So we have to identify the important features or descriptors for the particular biological activity. And this is done by several feature selection process. So after identifying the important features, we can go for model development. So models can be presented mathematically through equations, or the results can also be presented in a graphical way through contour maps. Some of the advanced machine learning models may not be presented through equations, because the simple linear relationship is not present in those cases. Whatever may be the case, after development of the model, we have to judge the quality of the model developed through different statistical tests. And when we are satisfied that the model developed is of appropriate statistical quality, then we have also to see whether the model is able to predict the activity of new set of compounds. So that is done usually by the process of validation. So once we are satisfied from the process of statistical quality valuation and validation, then only the model can be applied to new set of data. And the model can also be interpreted properly for mechanistic explanation. And the developed model can also be used for designing new set of chemicals. So if we want to see the workflow of any QSAR modeling exercise, we have to start with the biological data collection. And biological data can be obtained from different sources like online databases or literature, some scientific articles, etc. But we have to take care about the quality of data that is being collected. Because if the quality of the data is poor, the developed model will also be of no use. Then the collected biological data and the set of compounds should be subjected to biological and chemical curation. Now, as the data are being collected from different sources, the same compound may be present from in different sources in different names. So we have to omit those duplicates. And in some of the cases, the same compound may be present in different databases with different biological activity values. In such case, we may choose to take the average of biological activity value of the same compounds, or if the values are differing too much, then we are less confident that which value is correct, that we may omit some or all of the compounds. And similarly, we have to check for some structural normalization, and we have to check for some curation like removal of inorganics, removal of mixtures, and usually if there is some kind of compounds which is present in very low quantity, some specific structural features are present in a low number or quantity, so those dissimilar structures may be omitted before starting the QSAR modeling exercise. So curation is very much important. At the beginning of QSAR, and after collecting the data points and curating calculation, as I've mentioned, that so we can take help from different software for descriptor calculation, and there are different tools like AlphaDisk, Dragon, OChem, etc. for descriptor calculation. So after we calculate descriptors for the set of compounds that we have considered, we have to go for dataset division. The training set will be used for model development purpose, whereas the same be kept aside. This will not be used for model development. This will be kept as unseen dataset. So the feature selection and model development will depend on the training set only. The test set information will not be used. After development of the model, the developed model will be used for prediction of the test set data for which we have the experimental values, but we have blinded them. So the predicted values will be compared with the observed or experimental values, and when we see that the predicted values are close to the experimental values, that means residual is low, then only we can check the ability of the model to predict for the new set of data. So after we divide the data set into training and test, we have to go for feature selection using only training data. And this we can do using a number of methods, including genetic algorithms, stepwise selection, etc. And after we have selected the important features, we have to go for selection of appropriate learning or algorithms. Learning algorithms may be of two types. supervised and unsupervised. In case of unsupervised, we do not use the endpoint data or the biological activity data. So unsupervised methods are used mainly for uh, uh, checking the data and pattern recognition. This can be done by clustering or dimensionality reduction. Whereas in case of supervised model development, we use the Y data, and there may be two kinds of model development. One is regression, and other is classification. In case we have the quantitative biological activity values, we will go for regression model development. And in cases where we do not have quantitative values, but we have values like active, inactive, or high, low, we cannot use regression modeling strategy. We have to apply classification methods. So after we have developed our model, we will go for checking the quality of the model through different statistical tests, and then we will go for validation that OECD has provided a set of guidelines for QSAR model development and prediction and validation. So we have to abide by these guidelines. The first guidelines says that there should be a defined endpoint that we are going to model. So all experimental values that are used for QSAR model development should have been obtained using same experimental protocol. All the processes should be presented in the such way that it can be reproduced with ease by other group of authors. There should be a defined domain of applicability. So our developed model may not be applicable for whole universe of the chemicals. They are applicable only for specific kind of chemicals which are similar to the training compounds. So if the query compounds are much similar to the query compounds, to the training compounds, in that case, that particular query compounds may not be predicted with reliability from the QSAR model. The fourth guideline says there should be appropriate measures of goodness of fit, robustness, and predictivity, and these are done using various statistical tests and validation criteria. And the fifth guideline is there should be a mechanistic interpretation if possible. Now, according to the OECD guideline, we may show the QSAR work in this way. As per OECD guideline 1, we have to collect the data from literature having a particular endpoint, and then we go for the next steps, that is structure, representation, descriptor, calculation, and according to OECD principle 2, all the steps, so data pre-treatment and data set division, data processing, everything should be presented with clarity. Then according to OECD principle 4, there should be appropriate measures of the quality of fit and predictivity. So we have to use different metrics to know the quality of the model, and which may be or classification based model. So we have different kind of metrics, different metrics for regression and classification. Be able to predict for new set of data. The applicability domain of the model to know whether the query compounds are within the domain or not. And then, according to OECD Principle 5, there should be appropriate mechanistic interpretability of the model, if possible. Now, in general, the regression, that is statistical models that are used for QSAR model development are of two types: regression and classification. In regression model, the simplest ones are method linear discriminant analysis and logical machine learning models, which use artificial neural networks, support vector machine, random forest, and so on. Now, the metrics for judging the quality of QSAR models depends on whether you are using the regression-based models or classification-based models. In case of regression-based models, we have different kind of metrics like, say, determination coefficient or R square, And but the R square does not consider the number of features that are used for model development. So in such case, if we want to penalize a model for the number of features used, we have to use adjusted R square, where in the expression we can use the P, or the number of features that are used for model development. Then there are some error-based metrics like RMSE, which actually evaluate the degree of deviation between the observed and fitted values. And we have to take the square of the residual, and we take sum of the square of residual, divide by number of compounds, and we have to take square root. That gives RMSE. But these are these metrics that we have already mentioned, that is the R square, RA square, and the RMSE. These are just quality of fit. They do not say anything about the quality of prediction. So to know the quality of prediction, we have to use different kind of validation metrics. So there are different kind of validation measures like internal and external. So I'll come to those. So any QSAR modeling technique should ultimately lead to statistically robust models capable of making accurate and reliable predictions. So the QSAR model development can be divided into three stages: data preparation, data analysis, and model validation. The validation strategies check the reliability of the developed models for their possible application on new set of data, and competence of prediction can thus be judged. As I mentioned, the validation may be internal or external. In case of internal, we can do this by the method of leave one out, but we can also use leave many out. In case of cross-validation or internal validation, a segment of the training data is kept aside as internal test set, and from the rest of the compounds, the model is regenerated, and the left out samples are predicted from the regenerated model. And this is done in iteration. In the second iteration, some other set of compounds are kept aside, and from rest of the compounds, model is regenerated, and the left out compounds are predicted. In this way, we can find out the predicted values of different set of compounds. The simplest form, in each iteration we can omit only one compound, and this method is known as leave one out, but we can also calculate leave many out internal validation measures. Another method of internal validation is bootstrapping, where we omit some samples of compounds, and this can be done with replacement. So here subsamples are drawn in each iteration, and from rest of the compounds, model is generated, and the left out samples are predicted. And finally, you get the bootstrap R square value. In case of external validation, we do not use the training set data at all. We use the external set of compounds, and usually the external set of compounds, the additional compounds may not be available. So for addressing this issue, the original set of compounds is divided into first training and test set, as I have already mentioned. Test set compounds are kept aside, and after development of the model is used to predict the test set compounds. And then the predicted values are compared with the experimental values, and if the predicted values are close to the experimental values, then the model is good, and this is quantified in terms of R square trade. As you can see the expression here, the value should be close to 0.6 or higher. Now in case of classification, we cannot use the measures like Q square LO or Q square F1. We have to use different kind of metrics like sensitivity, specificity, accuracy, and precision. Those are derived from true positive, true negative, false positive, and false negative from contingency table. And we can derive F measure, Matthews correlation coefficient, and we can also judge their quality from the receiver operating curve, or ROC. Now, if we see the application of QSAR, QSAR is applied in pharmaceutical science for lead optimization and drug development, ADME optimization. It can be used in material science for optimization of different properties. It can be used in food science, agricultural science, nanoscience, predictive toxicology and for regulatory aspect. Now, another related data gap filling technique is read across, where QSAR is a statistical method. Read across is not a statistical method. It is based on hypothesis and is based on the similarity principles. So for the compounds without the experimental values, or for the query compounds, we have to identify its close congeners. And the definition of closeness may be based on the structural similarity, but we can also include other kind of similarity, like similarity in the metabolic profile or similarity in toxicity, etc. So based on defined similarity, we have reduced uncertainty. We have to give argumentation and justification how we define the other compounds as close compound to a particular query compound, and which respect. And in this way, the confidence may be grown, and based on that, the hypothesis may be accepted for regulatory decision. So this is a purely non-statistical approach, but one can place this in a statistical modeling framework. So we have recently developed a framework of quantitative read across, where we put it in a quantitative framework. So starting from the data set, the data set is divided into two parts: source and query compounds. The source compounds are like training, and query compounds are like test. And the set of source compounds are again divided into sub-training and validation set. And the validation set is used for optimization of different similarity criteria, for hyperparameter of different kind of similarity in Euclidean distance-based, Gaussian kernel-based, and Laplacian kernel-based similarity definition. So once these hyperparameters are optimized from the validation set, we can use these optimized hyperparameters for prediction of query compounds to chemical read across through weighted average predictions. So once we get the predictions, then we can apply the usual validation metrics as are used in case of QSAR. Now, to give example, say we have a query compound like bromobenzene, and we want to know the endpoint value for bromobenzene from the similarity principle. And say, for example, we have identified some of the close congeners like say toluene, chlorobenzene, and phenol. So we have to see we have to check the similarity of bromobenzene with the close congeners like toluene, chlorobenzene, and phenol. And based on that, we will define kind of similarity, Euclidean distance based, Gaussian kernel based, and Laplacian kernel based similarity. And based on this similarity definition, using these similarity values, we will calculate the endpoint value through weighted average prediction for bromobenzene. In this way, the read across prediction can be done for different compounds, and once we get the prediction, then the predicted values can be compared with the experimental ones, as we do in case of QSAR. Now, as I mentioned, the QSAR is a statistical approach, so it requires a good number of compounds for statistical reliability, because it needs a good degree of freedom for reliability. So when the number of compounds is very limited, we may not be able to apply QSAR to generate a statistically meaningful model. So in such case, read across may be useful, because read across does not require statistical model building. On the other hand, QSAR can give quantitative information for different features, which may not be possible in case of read across. So we can take the merits of both of the methods, QSAR and read across, and we can combine them in the same framework, and that is called read across structure activity relationship. So this is the workflow for our quantitative read across structure activity relationship. So here we have two set of compounds. One set is training, and other set is query. But we divide the training set into sub-training and validation set, and we define different kind of similarity measures, and we optimize the read across similarity measures through prediction from the validation set. And once the optimized setting are obtained, we calculate read across structure activity relationship descriptors. So these are basically similarity and error based descriptors. I'll come to this And once we calculate the rasa descriptors, we'll combine them with the usual structural and physiochemical descriptors. And on the combined set of descriptors, we will apply feature selection. I will develop the model, and from the developed model, we will get the prediction from the test set compounds, as we do in case of QSAR. But RASA is different from QSAR in some respect. I'll try to explain here. Now, say we have a set of compounds, and obviously different compounds will have different kind of biological activity values. So some compounds will have low values, some compounds will have higher values. So in the training set, we will have a mean of the training set response. So if we consider that mean as the threshold, so we can classify the training set compounds into two parts. So one set of compounds with values higher than the training set response mean may be considered as positive compounds, and the rest of the compounds having the response value lower than the training set response mean may be considered as negative set of compounds. So in this way, we can classify them. And for each query compound, we can identify their close compounds, and based on the activity values or response values of the close compound for a particular query compound, you can calculate different error methods like standard deviation, their coefficient variance, and standard error. On the other hand, we can identify, we can calculate the similarity of closed source congener for each query compound using different methods like Euclidean distance, Gaussian kernel, and Laplacian kernel, and we can calculate the similarity values. And from that, we can calculate average similarity, HD similarity, and CV similarity. Now, depending on the positive or negative compounds present in closed source congener, we can also calculate maximum positive, maximum negative similarity values and their absolute difference, and positive average similarity and negative average similarity, etc. So these metrics are actually used, these measures are actually used as RASA descriptors, and from that we can also get different derived that are shown at the bottom of this slide. I will not go into details. So this shows the basic RASA descriptor list: SD activity, CV activity, average similarity, SD similarity, maximum positive, maximum negative, absolute difference. And we also use a concordance measure G based on the value of positive fraction, that is the fraction of the compounds present in the closed source congeners for a particular query compound. Now, G or concordance measure has some ambiguity depending on its closed source congeners and the positive fraction values, because for the same set of positive fraction value, it may show same G value for different sets of positive fraction values. So, for example, the G value will be 0.8 for when the positive fraction is 0.6 and also negative fraction 0.6. So we can address this ambiguity by defining another measure, that is GM. I'll come to that afterwards. Now we have also developed another composite function which is called RA function, which takes into account all contributing descriptors through the similarity definition, and we must combine the RASA descriptors, that is similarity and error based descriptors, with the usual structure and physiochemical descriptors for getting more precision in the prediction. And the third the third picture here shows different scenarios where for a particular query compound which is shown in the form of a question mark, the closed source congeners may be of different types, where most of the compounds may be positive. That means positive fraction may be 1 and GM value will be 1, or the most of the close congeners may be negative, so maximum positive will be less than maximum negative and GM will be negative, or they will be in the borderline where the close congeners may contain both positive and negative compounds. In this case, GM will be 0, so reliability of for such compounds will be low. So according to Rasser algorithm, we start with the chemical set and we get the feature vector in the form of similarity based on fingerprints or some other properties may also be possible. And And we apply supervised learning technique for more prediction for multiple endpoints, and based on similarity, maximum similarity of the query compound to known inhibitors or different targets, we can link them to different molecular initiating events that are relevant for adverse outcome pathway. So RASAR algorithm has also a possibility to link to the adverse outcome pathway. But in the simple form, if you want to describe RASAR, which is a combination of QSAR and read across, starting from a data set, we compute 2D descriptors, but one can also compute 3D descriptors. And we divide the results into training and test. We select the features, we calculate, we develop the QSAR model. At the same time, we calculate the similarity measures and combine them with QSAR features. We develop the RASAR model by combination with similarity different from QSAR in several respects. In case of QSAR, we calculate descriptors for a particular compound from the structure of that particular compound. Whereas in case of RASAR, we calculate RASAR descriptors not from that particular compound, but from its closed source congeners. Secondly, in case of QSAR, we calculate descriptors first, and then we go for training and test after division of the data set into training and test, because here test set compounds are used as query. So the RASAR descriptors are calculated by calculating similarity of the query compounds to different training or source compounds. So in this way, RASAR is different from QSAR, and as we calculate RASAR descriptor based on the closest congeners, so the prediction aspect will be inbuilt in get that the external validation quality for RASAR models are better than QSAR. Now we have performed several case studies. I'll give some examples here. The first one is from androgen receptor binding affinity. This is a collection of rat androgen binding affinities with 200 data points. So after structural curation, descriptor calculation, data pre-treatment, we go for data set division. Finally, we have a training set of 103 compounds and test set of 44 compounds. We select our features through genetic algorithm and followed by base subset selection. And finally, we develop our QSAR model based on partial least squares. And in the right hand side, we have shown the model. As you can see, there is a mathematical equation. There is equal to sign. The left hand side, there is log RBA. This is the Y value or dependent variable or the receptor binding affinity that we are modeling. But in the right hand side, you have several descriptors like S triple SCH, max A, CH, etc. So these are actually the information about the molecular structure, and we can see each descriptor is associated with a number with either positive coefficient or negative coefficient that describes the contribution of that particular descriptor towards the response. And followed by these, we see different kind of measures or metrics, like say the R square train, as I already mentioned initially, the determination coefficient, then the Q score LO, that's the internal validation quality or false validation measure, and Q score F1, that is the external validation measure, the value of which should be more than 0.6, but here we are getting 0.58 in case of QSAR model. So this slide also shows that the quality, that the source of the data is the KDB database. Then we have the 102 training compounds and 44 test compounds, and the same equation is shown here, where we find that Q square F1 value is 0.58, which is lower than 0.6. And these are the descriptors which are used during QSAR model development that describes different structural features, but I will not go into details here. Now, when we apply chemical read across, which is the non-statistical approach instead of QSAR, we see that Q square F1 is in private, we get here 0.635. So using the optimized hyperparameters, we get the better quality of predictions. This is followed by RASAR model development. As I mentioned, that RASAR is a combination of read across and QSAR, where we use the QSAR descriptors or structural physicochemical descriptors along with similarity measures. So we use both the QSAR and raster descriptors in the same model. So we get here 4 individual models and some pooled model, and also we get for consensus. Consensus means different consensus prediction in this work. Now this slide shows the bubble plot for different descriptors. So here the bubble size represents that is actually the reflective of the importance of individual descriptors, and we will see that the RASA descriptors, which are having the more bubble size, so RASA descriptors are more important than QSAR in most of the cases. This graph shows that the highest external validation in terms of Q square F1 is obtained from pooled descriptor RASA models. So pooled descriptor RASA model shows Q square F1 value of 0.671, superseding both QSAR and rate across. And in case of, if we take the MAE value, that is the mean absolute error, which should be minimum, that is obtained from the consensus model in RASA model, and this is superseding the quality of model of QSAR and rate across. Now, one more interesting thing, I showed one measure, that is G, or concordance measure. I told that it has some problem. So in the first plot where G has been used, and the BIP plot shows that the G is not much important. But when G is replaced by GM, the modified G, the expression is shown here, it shows the second highest importance. So GM is more important than G. So we use GM in our work, modified G instead of G. In the next work, we have used RASA for machine learning based QSAR prediction of different toxicity endpoints. Here we have used rat androgen receptor binding affinity, acute oral toxicity data in avian species, specific avian species, acute contact toxicity in honeybee, acute oral toxicity in Melartic and avian species, and inhalation toxicity data. And this also shows the bubble plot showing that RASA descriptors are more important in most of the cases. Now, this is a comparison of model quality for RASA and QSAR. The first row shows RASA, the second row shows QSAR, and this is replicated in different datasets. As you can see, the Q square F1 is the external validation measure. For dataset 1, Q square F1 is 0.70 versus 0.58 for QSAR model. So RASA model is superseding the quality of external validation for dataset 1. This is shown in all the dataset. In case of all the dataset, the QSAR model shows the better quality of external validation than QSAR models. And this different scenario that we can obtain, our query compound may be close to the compound and their activity
Ma'am, your voice is not clear. Sir, your voice is not clear.
Maximum positive value negative will be negative. They are close to, and their GM value will be close to 0. This
Actually, Professor Roy has some connection problem. He will be joining soon.
I have joined soon. Join again.
Okay, thank you, sir.
It was disconnected just
Sir, again your voice is breaking.
now. It was disconnected just now.
So, sir, your voice is not clear, breaking. Can you please disconnect and rejoin?
Yes, sir. Yes, sir. Voice is not clear.
Hear me now?
Yes, sir. Yes, sir.
Yes. Now
Okay, okay,
audible.
okay. Okay, so this is RASAR modeling for hepatotoxicity data set. And after collection of the hepatotoxicity data, we do appropriate structure representation, descriptor computation, and data pretreatment, then data set division. So the training set is used for model development, test set is kept aside. This is not used for model development. And feature selection is done solely from the training set. After selecting the features, we do the appropriate similarity descriptor computation. And from these raster descriptors, we again select the important features and we go for machine learning model development. LDA, that is linear discriminant analysis, logistic regression, random forest, and support vector machine. So this we do for both QSAR and RASA. And then we compare the quality of the test set prediction for QSAR and RASA. As you can see, that if we consider Matthews correlation coefficient, that shows that the QSAR model, the logistic regression shows the MCC, 0.22, that is the highest value. Whereas in case of RASAR, for the test set prediction, logistic regression gives 0.27, whereas LDA gives 0.29. This is the highest value. So the consideration. Now for doing this, for the set of compounds, we can divide them in two parts, positive and negative, based on the training set mean. And we can normalize the selected descriptors, and we can calculate the average values of those normalized descriptors for both the classes, positive and negative. And then we can find whether the descriptor values are showing high, that is the higher value for the positive class or negative class. So some of the descriptors will show higher value for the positive class, some of the descriptors will show higher value for the negative. And we can calculate weightage based on the formula that is shown at the bottom of the slide. That is the difference of average values for the both classes and divided by sum of different values of all descriptors in a particular class. And then we multiply these weighted values for this with the standardized descriptors, and we can calculate ORCO descriptors, arithmetic residual in K group analysis in two dimensions, ORCO 1 and ORCO 2. Now, when we plot ORCO 2 versus ORCO 1, the positive component should reside in the fourth quadrant and negative component should reside in the second quadrant. But when the opposite happens, that is, if negative components are present in the fourth quadrant and positive components are present in the second quadrant, where the closed source compounds are of opposite class, but a particular shows in the unexpected class. So when we find small changes in the activity in the features, the activity value too much, then we can find out activity claims. So this can be easily identified using this approach by calculating all descriptors. We can give some examples like say bupivacaine, which is a close congener, is non-heterodoxic. This is an example of activity claim. Similarly, dianosterol is non-heterodoxic, whereas phenol is heterodoxic. This is another example of activity clip based on their close similarity value. Again, amphotericin B is heterodoxic, whereas its close source congener nystatin is non-heterodoxic. This is another example of activity clip. Esomeprazole is non-heterodoxic, whereas is most close. So this and we have one hyperparameters, and for different machine learning algorithms we have different tools which can be downloaded from our site. For the of our site is given on we have also applied raster for different other endpoint values in material science and toxicology, etc. So this slide shows application of machine learning based QRASPER approach for efficient prediction of specific surface area of perovskites, inefficient prediction of cytotoxicity of titanium dioxide based multi-component nanoparticles using machine learning based QRASPER approach. So these studies report the development of simple, interpretable and reproducible QSAR models for various toxicity and also activity and property endpoints. The QSAR models reported here thus deliver lower prediction error for the query sets than the corresponding QSAR models, suggesting that they are potential models of choice for efficient prediction of a given level of chemical information. So based on the variable important plot, in most of the cases we have seen that RA function, GM and average similarity are the most important similarity-based determination for prediction of toxicity, which warrants further extensive studies on this function. The ORCO descriptors may help to identify activity cliffs, and the combination of ORCO and with raster can give a good modeling strategy based on which we have recently modified the raster workflow, and the second generation raster we use the ORCO descriptors for calculating the similarity basis. As raster is essentially a similarity based approach, identifying activity cliffs may help the raster models effectively. The use of raster and ORCO approaches in may prove to be an effective strategy for predictive modeling. Research were funded by Science and Engineering Research and Development Organization. But we also acknowledge the funding received from a book on QRASPER, published as Stringer Brief. There are several other books on QSAR and their application. Is the QSAR with application in different fields. This is Springer Brief. This is a reference book, Alzheimer, on our website, from which this can be easily downloaded and used. So this is the QR code, and the site address is also given here. Thank you very much for patient hearing. So if you have any question, please feel free to ask.
Thank you so much, Professor Roy, for your insightful and informative talk. Now, dear participants, I have a small message to all. Please remember there is no such thing as a silly question. Every question is a step toward learning something new. So, let's begin the question and answer season. Please feel free to ask your questions, clear your doubts, and you have any ideas, please share it. Dear participants, if you have any questions, don't hesitate in asking questions.
You can ask in the chat box also, but in the chat
Yeah,
box I see only thank you, but there is no question.
you can raise your hand, you can type the question in the chat box, or you can directly unmute your mic.
Yes. Chandran Manikam, raised hand.
Dr. Chandran, have you any questions? You have raised the hand. I think he has no any question. Any other participants? There is a question, I
There
think,
is some
from
message
Lovely
here.
Raj.
A,
Yeah.
a collaborator gives a data set of compounds and asks to predict their toxicity. Which class of molecular research would we prioritize? That depends on the kind of data that has been given. That cannot be predicted beforehand. So usually the descriptor computing software can compute several kind of descriptors, and one has to go for the process of feature selection. So after feature selection only one can be sure which kind of descriptors will be of use, but based on the kind of particular type of activity or toxicity some activities are based on the hydrophobicity. Some activities are based on, say, steric features. Some activities are based on the electronic features. So there may be some requirements of specific categories of descriptors. For example, if we say you dealing with some activity which is based on the electronic phenomena, so you must use some electronic descriptors. Uh, so in that way, otherwise one should calculate different type of descriptors, and one should go. So prioritization will depend. Also sometimes some activities are based on some 3D representation, 3D information, especially the pharmaceutical activities. So in those cases, the 3D structure consideration is important. Otherwise, for toxicity values, in most of the cases, 2D representation is sufficient.
There
For
is one more.
collinearity, one should go for dimensionality reduction, and one should use techniques like the partial least squares, which can handle the method of that is the problem of collinearity. So one can use partial least squares instead of a Miller. that can deal with the problem of collinearity, or one should go for some dimension reduction method like PCA, principal component analysis, and one can use the components as descriptors.
Sir, is there any role of threshold?
Threshold, response threshold
Yes, sir.
for classification, for classification modeling. For which kind of modeling? Because in case of regression based modeling, usually threshold is not used except in case of RASA. But if you are doing classification, then a definite threshold will have role in your model quality. So if you use different thresholds, you will have different model quality. If you know some reference compounds which can be used as threshold, so that particular reference compound can be used. Otherwise, your training set response mean can be used as threshold.
Thank you, sir.
Any other questions for participants? So I think there are no any questions. Thank you so much, sir. Thank you so much for your amazing talk in silico toxicology. And you made such a complex topic so clear. And I think even our computers are taking notes. So we truly appreciate your time and the valuable knowledge you shared with all of us. Thank you. Thank you very much.
Okay, thank you very much. Thank you.
All right, everyone. So that is the end of fifth day of our FDP. But let's heads up for tomorrow. For tomorrow, it's going to be a long season, starting at 2 p.m. and going till 8 p.m. Yes, I am saying right. Tomorrow will be a 6 hour marathon. So make sure you get plenty of rest tonight. And since tonight is Friday night, I think it's a weekend night. So go ahead, enjoy a little party, chill, watch your favorite show. But just don't forget to recharge because we need you back full of energy and ready to learn tomorrow. So see you all tomorrow at 2 p.m. sharp. For now, good night, sleep well, and dream of all the knowledge you will gain tomorrow. Good night, Shubh Ratri, Shabba Khair.
Shubh Ratri, sir. Shubh Ratri. Thank you so much.
Thank you, sir.